Spot plates are used to enumerate the concentration of bacteria in liquid media through serial dilutions, typically in the ratio of 1 to 10. We always start by sterilizing the workbench with 70% ethanol, and then lighting the Bunsen burner. Collect a cell culture sample from your chemostat. You won't need more than 5 milliliters. Then vortex to create a homogeneous solution. Our cell culture sample is Pseudomonas aeruginosa. In order to make a spot plate, we are going to need a liquid culture sample from the chemostat, a 200 microliter multi-channel pipette and tips colored green, a 20 microliter multi-channel pipette and corresponding tips, which are colored red, a deep well, a 96 well microtiter plate, and 2 agar plates with 2 strips of parafilm. And lastly, a couple milliliters of sterilized MD media spiked with glucose. Start off by labeling the agar plates, indicating which will be the blank and which will be the spot plate. Always include your initials, the date, and the type of organism being plated. The 96 well microtiter plate has 12 columns labeled numerically from 1 to 12 and 8 rows labeled alphabetically from A through H. The 96 well plate comes with a lid which should be used to cover the plate to maintain sterility and can help keep track of which row or column you are pipetting from. The spot plate is begun by taking the OD600 reading of triplicate samples of the initial stock of Pseudomonas bacteria. We take the OD600 to draw a relationship between the OD and the concentration of bacteria. First, we are going to take 200 microliters of the stock solution and pipette them into wells A1, A2, and A3. Here we are using the 200 microliter pipette to pipette from the sample. Remember to keep the tip box closed when not in use to maintain sterility. After pipetting 200 microliters of the sample into A1, dispose of the tips and the biological waste. Again, using sterile technique and working close to the flame, pipette 200 microliters into A2 and A3. Always dispose tips that have bacterial contamination in the biological waste. Now we should have triplicates of the Pseudomonas stock solution in A1, A2, and A3. Keeping track of our pipette work, our plate should now look like this, with 200 microliters in the top 3 wells. Now we are going to read the OD600 in the Biotech plate reader of the top 3 wells. After turning on the Biotech plate reader, be sure to take the lid off of your plate and align the A with the corresponding A1 in the top left corner of your plate. We will use the Gen 5 software to read our plate. Select the wavelength we want to read at. In our case, it is 600. After hitting run, we can achieve our OD600 values. Disregard the values from A4 through A12. We only care about the readings for A1 through A3 where we put stock solution sample. Now we are going to use a 200 microliter multi-channel pipette to pipette 180 microliters of MD media into columns 1 through 3's, rows B through H. Remember to use sterile technique and flame the cap and the mouth of the bottle when opening the MD media to pipette from. We are going to fill the first 3 columns of our deep well with MD solution so we can easily access MD media with the multi-channel pipette. I use the automatic pipette to quickly grab large volumes of the MD media to fill up the deep well. Once the deep well has the first 3 columns filled with MD media, we can now use 3 tips on the multi-channel pipette to pipette into the triplicates with ease. After the 200 microliter multi-channel pipette has been set to 180 microliters, grab 3 tips. Then using the deep well filled with MD media, pipette 180 microliters into row B's triplicates. Continue to pipette 180 microliters through row C through G. Tips do not have to be discarded between rows here because no bacteria is being used. We are also taking caution not to touch the rim or any other parts of the 96 well plate. Finally discarding the tips, our plate should now look like this. With 200 microliters of stock bacterial solution in A1, A2, and A3, and 180 microliters of MD media in the other wells. The next step in the serial dilution is to transfer 20 microliters of stock solution from row A into the triplicates of row B. This will create a 10 to the minus 1 dilution of the stock solution in row B. With fresh tips, we're going to repeat this step and now transfer 20 microliters of solution in row B into row C to create a 10 to the minus 2 dilution of the stock solution. Continuing and repeating this step throughout the plate until we hit row H, we are going to keep increasing the dilution factor by a factor of 10. The last 20 microliter transfer from row H will be discarded, so that all wells contain 180 microliters of solution. Here we will use the 20 microliter multi-channel pipette and corresponding tips. After ensuring that the pipette is set to 20 microliters, grab 3 tips to transfer the triplicates of row A to row B. After dispensing 20 microliters of the stock solution in row A into row B, pipette up and down about 10 to 15 times to ensure that the concentration of row B's triplicates is homogeneous and at a dilution factor of 10 to the minus 1. Then dispose of your tips. Grab new tips and transfer 20 microliters from row B to row C and pipette up and down 10 to 15 times to make sure row C is now at a uniform concentration. Then dispose of your tips. Now with fresh tips, we are going to do the same procedure for row C to row D and transfer 20 microliters from C to D. Again, pipetting up and down 10 to 15 times to make sure the new concentration of D is uniform and disposing of the tips afterwards. 20 microliters will be continued to be transferred throughout the rows until we reach row H. Since serial dilutions are concentration dependent, disposal of tips between rows is mandatory. The lid of the microtiter plate can be used as a marker to indicate which row you are working on. Be sure to be diligent and not lose track of which row you have transferred your serial dilution into. The final 20 microliter transfer from row H is discarded. The microtiter plate will now contain 180 microliters in all wells with the most concentrated solution in A, which contains the stock solution of the bacteria. Here's a diagram of the dilution series we have created. With the most concentrated solution in row A with the original stock of bacteria and the least concentrated solution in row H with a 10 to the minus 7 dilution factor. Now we are going to plate the dilution solutions we just made onto an agar plate using the 20 microliter pipette. We are going to set it to 10 microliters and insert 7 tips. Now move the lid to grab only from sample 1. We are going to grab from row B through H, the dilution series from 10 to the minus 1 to 10 to the minus 7, and very carefully plate it onto the agar plate. Very slowly and carefully eject the cereal dilutions so that they remain in drop form. Then eject your tips. Using a fresh set of tips, we can also plate 10 microliters of sample 2 and sample 3 onto the same plate, very carefully so that our cereal dilutions are parallel. Be careful not to move the plate so our dilution series do not run into each other. The most heavily concentrated spots will be towards the top with a dilution of 10 to the minus 1, and the least concentrated will be towards the bottom with a dilution of 10 to the minus 7. Leave the Petri dish open to dry. It may take more than 20 minutes. When you can no longer see the spots, close the Petri dish, parafilm, and place upside down in the 37C room to incubate. Now that we are done with the chemostat liquid culture, we need to inactivate it. Use the disinfectant 100% bleach and pour just enough to make it a 10% bleach solution. The Pseudomonas liquid culture sample can now be disposed of in the biological waste. After the spot plates have incubated for 24 hours in the 37C room, bacteria should have grown. Now to enumerate the bacterial concentration, start from the top, the most concentrated. Countable bacterial colonies are anywhere from 10 to 80 colonies, and for us this seems to be on row 4. Take note of the dilution factor that you are counting your countable colonies at. Also, remember that we plated 10 microliters of solution onto the plate, and then use the OD600 to go back and back calculate the initial concentration of the stock bacteria.